A very warm welcome to today's class. Today we will be dealing with a very interesting topic, which is embryonic development. Why I said it is interesting is because today we will see how a single celled zygote forms a multicellular organism that is fully functional. So, we will deal with all the developmental changes that are taking place from the zygote till a complete human being is formed, and the entire procedure, whatever changes takes place during these period are called as embryonic changes. So, students, today we will deal with some of the major physiological changes and anatomical changes that are taking place. So, let us start with what the changes are and how we can broadly classify the different aspects of embryonic development. So, we can study this entire concept of embryonic development following these four particular criteria. First, its cleavage, then the growth, morphogenesis and differentiation. एगुड़ाक कौन आमे डिटेल्स रे डिस्कशन करिबा. So, let us start with what is cleavage. अच्छा पिला माने जेते बेले गोटे सिंगल सेल्ड zygote, समस्ते जानिच zygote is a diploid cell and it is formed by the fusion of ova and sperm, which are your haploid gametes, and this particular process ame last class re padithile, it is called as fertilization, and after fertilization the end product is your zygote. Now, how this zygote will develop into a complete human being? This zygote will undergo certain changes. What are those changes? Those changes are cleavage, then growth, then morphogenesis and differentiation. अच्छा, सो हमें चारिटा एस्पेक्ट देखिले, क्लीवेज, ग्रोथ, morphogenesis and differentiation. What are all this? Let us start with cleavage. Now, cleavage is a very rapid mitotic division. कौन हुए एथरे ना मोर zygote टार जहाँ size अच्छी, the size remaining same, it will rapidly divide into different type of cells and it will form a blastula. Up to the formation of this blastula, we say that this process is called as cleavage. कौन हुए? Now, this rapid division takes place, but there is no growth at all. And देखि पारुथिबो एठी zygote size जहाँ अछि, each stage after division size is also remaining the same. It is just that एठी तमरो गोटिए nucleus थिला, here it is dividing into 2 cells, here into 4 cells, first 3 cells, tapare 4 cells, then it will come to say 8 celled, then 16 celled, then 32 celled and so on. So, only cellular division is taking place and here there is no growth. Tapare kon asibo na growth phase. Once we reach this particular blastula phase, it is a constrained unit where there is no growth, but only mitotic division. At this stage, this blastula has to be implanted in the uterine cavity, and for this purpose there has to be certain changes that are accompanied by growth. What will happen in growth? In growth, the size will increase. It is no more constrained now, the size will start to increase. Now, whatever daughter cells were formed, they all will grow in size. Thereafter, I said morphogenesis. Morpho means form. Okay. So, morphogenesis means when certain cells will migrate, certain movement will take place, certain movements when they will take place that will shape your embryo, shape the embryo, those are called as your morphogenetic movements, and this process is called as your morphogenesis. आमे इथरे इन्वेजिनेशन केमती होची, एपिबोली कोण, जर्म लेयर फॉर्मेशन केमती होची, सब डिस्कशन करिबा इन डिटेल्स. आफ्टर मॉर्फोजेनेसिस व्हाट विल हैपन, मॉर्फोजेनेसिस रे हमरो एम्ब्रियो रो गोटे डेफिनाइट शेप आसी गला. नाउ वंस वी हैव अ डेफिनाइट शेप, नेक्स्ट कम्स फंक्शनल एस्पेक्ट. डिफरेंशिएशन रे कोन हबो ना, जो पर्टिकुलर fixed orientation or the shape of the embryo that has been attained by morphogenetic movements, they will now differentiate into different functional categories. Suppose more got a group of cells, there are many group of cells in my embryo, certain group of cells will form my digestive system, certain group will form the neural tissue or the nervous system, so on and so forth, like different systems and different functional aspects of the embryo will develop. That is what we will deal with in differentiation. ताले चारिटा different aspects of embryonic development clear हैला. First रे cleavage हबो, जो रे खाली division हबो, growth हबो नाही. Second रे जहाँ cleavage है कि divide करथिला cell गुड़ा को, they will all grow in size. Third, these growth will be accompanied by morphogenetic movements. Okay, movement of those particular cells in particular orientation to give a definite shape to the growing embryo. Now, once my embryo has got the shape, differentiation of these cells will take place and they will be functionally active. ताले entire process of embryonic development रे starting from one single cell, we will get a functionally active individual that is formed. ताले चलो देखिबा details रे केमते सबु process गुड़ा को human body रे होची. So, coming to the entire embryonic development, before going to the process, आउ टिके discussion हमें इठी करिबा जे पूरा embryonic period we can divide it into 3 parts. तम माने सुनिथिबो fetus, embryo, there are different terms accompanied and बहुत थर यही terms गुड़ा करे confusion भी हुए, fetus कहा को कहिबा, embryo कहा को कहिबा, तो सेही सब confusion ए part रे clear ही जिबा. This entire embryonic development can be divided into 3 phases. First is my pre-embryonic development, then embryonic development, तापरे fetal development. Pre-embryonic development कोण, जोटा हमें zygote पढ़िले, से जो zygote formation हबा परठू, that is starting from fertilization till to 2 weeks of development को हमें कहिबा pre-embryonic development, or we can also say it is also called as germinal development. Okay, it will begin with fertilization and obviously तमरो fertilization कोठी होची ना, ampullary portion of your fallopian tube, one of the fallopian tube, जोटी तमरो fertilization होची. So, the site of the initiation of this germinal embryonic development is the fallopian tube. Now, the zygote will travel to the uterus. After being formed, it will travel to the uterus and it will attach itself to the uterine endometrium, जोटा हमें fertilization पढ़ला बड़े भी पढ़ी थिले. Now, after it will attach, rapid cellular divisions will take place and this single cell zygote will form a group of cells. Once the zygote is successfully attached to the endometrial lining, हमें एठी germinal stage टा को end करिबा, or your pre-embryonic development will end when the zygote has attached to the endometrial lining of the uterus. तापरे कोण germinal or pre-embryonic गला, pre-embryonic पर हमर असला embryonic life. Embryonic life हमें केतेबेले कहिबा when it is the development that is taking place from 2 to 8 weeks after fertilization. ताले जेतबेले embryo टी develop करि सारिची, 2 weeks जाए divide करि सारिची, आउ uterine cavity रे जाके attach हेई सरला, तापरे till 8 weeks जहाँ changes हैला सेटा को आमे कहिबा embryonic development. The developing embryo will form all the major systems, organs and structures within these 2 weeks. एटा को आमे organogenesis भी कहिबा. First 2 weeks रे organogenesis हुए and this will begin when the zygote will attach to the uterine endometrium. And केते बळे एटा को period जाय limit करिबा आमे this activity or this 2 to 8 weeks रो period टा सेठी सरीबो जते बळे हमरो ossification start हबो. Bone formation हबो. Bone formation जते बळे first ossification आरंभ हबो आमे सेठी embryonic development टा को limit करिबा. तेनु 8 week जाय आमे ताको embryo बोली कहि परिबा. Okay, is it clear now? Up to 8 weeks we can say the developing zygote to be a embryo. Once 8 weeks of development has been completed, तापरे आमे ताको कोन कहिबा? We will say that it is a fetus. Fetal development is from 9 to 38 weeks. Okay, from 9 weeks 8 weeks परे जहाँ start हैला 9th week start हबो, so 9 through till birth we can say it is at 38 weeks or 48 weeks after fertilization. So 48 जदि पूरा stretch टा को धरिबा ताले it is 40 weeks or if after fertilization I mean 2 weeks minus कर देबा it is 38 weeks. So from 9 to 38 weeks or till birth we say it is fetal stage. Now fetal stage रे कोन हबो? Fetal stage को आरंभ होइथिला कोठु जोठु ossification आरंभ होइथिला. So that marks the starting of the fetal stage. 8 weeks सरी गला माने आमे ताको embryo कहिबानी. 8 weeks परे आमे ताको कहिबा it's a fetus. Now this fetus will experience rapid growth and further development of all the body structures, organs and system. 0 to 2 weeks भितरे organogenesis हेइथिला. तापरे 2 to 8 weeks भितरे तमरा सबु organs गुडा को जहा formation हेइथिला those have developed in the embryonic system, embryonic period, and at the fetal period there will be rapid growth of all these organs that were formed and the system will be well equipped to face the after birth sequences. ए डायग्राम टी रे तम माने देखि पारिबो आमे जो तीनटा पार्ट रे डिवाइड करले पूरा एम्ब्रियोनिक डेवलपमेंट को. फर्स्ट इज योर germinal development which is from 0 to 2 weeks, embryonic which is from 3 to 8 weeks. Embryo has been formed and this embryo takes the shape of, it looks somewhat like a human, a miniature human with placenta formation and limb buds देखि पारथिबो त माने डायग्राम रे न्यूरलेशन हेछि and then the fetal development start after 9 weeks till the birth. ताहा परे आमे जिबा क्लीवेज को. जहाँ सब डेवलपमेंट आमे देखिले from starting from the formation of a zygote till birth आमे तीनिटा भागरे embryonic period को divide कले. Now coming back to the basic of the developmental changes. First आमे discussion करथिले cleavage, morphogenetic changes and differentiation. ताहले देखिबा cleavage कोन. Cleavage is, we can say in human beings it is holoblastic and indeterminate cleavage. ताहले देखिबा holoblastic आउ indeterminate आमे काहा को कहिबा. Cleavage मु आगुरु कहि सरछि it is a rapid mitotic division without the growth of the cell. ताहले rapid mitotic division जते बळे होछि हमरो normal cell गुडा करे G1 and G2 phase रोछि जोथरे growth भी होछि किंतु एई केस रे हमरो growth phase नाही. ताहले cleavage रे कोन होछि rapid mitotic division without growth. Suppose this is my single cell and then it forms two celled. Then this two celled will form a 4 celled structure. And then there will be a division to form a 8 celled structure. एई जो division गुडा को पिला माने मु blackboard रे कली, these are of two types. गोटे जगह रे देखिलो एई axis रे division हला, आ गोटे जगह रे देखिलो एई axis रे division हला. एटा को हमें कहिबा this is vertical cleavage. Okay, and this we can say as horizontal cleavage. ताले दीटा प्लेन रे हमरो क्लीवेज होची और डिवीजन होची. जेतबेले वर्टिकल क्लीवेज होची वी से दिस एज मेरिडियनल क्लीवेज एंड जेतबेले हॉरिजॉन्टली होची वी से दिस एज इक्वेटोरियल क्लीवेज. ह्यूमन बीइंग केस रे कौन हुए ना रोटेशनल क्लीवेज हुए. व्हाट इज रोटेशनल क्लीवेज? सपोज मोर फर्स्ट ए एक्सिस रे क्लीवेज हेची, नेक्स्ट माय क्लीवेज विल बी एट 90 डिग्री. ताहले सेल गुडा को गोटे ऊपर गोटे स्टैक हेकी रही जीवो. Unlike the other animals where different kind of cleavages takes place in the entire animal kingdom, but एठी कौन होबो ना हमर जत्तेबेले भी क्लीवेज होची there will be stacking of cells one above the other. ताहले रोटेशनल क्लीवेज आमे ए टाइप रो क्लीवेज को कहबा जोथरे गोटे एक्सिस रे जदि फर्स्ट क्लीवेज होची सेकंड will follow the next axis which is perpendicular. ताले क्लीवेज रोटेशनल कहीं बोली आम जानले. एबे देखिबा होलोब्लास्टिक क्लीवेज कौन. होलोब्लास्टिक क्लीवेज माने सपोज माय zygote has been divided and it has now formed a group of cells, be it any group of cell. If all the cells are taking part in the cleavage, if all the entire cell is taking part in the cleavage, then we say it as होलोब्लास्टिक क्लीवेज. आउ कौन आम या कहे वी से इट इज indeterminate cleavage. What is indeterminate cleavage? Indeterminate cleavage. Indeterminate माने suppose before implantation मोर ब्लास्टुला फॉर्मेशन हो गला, ब्लास्टोसिस जेंकी इम्प्लांट होबो यूटेराइन एंडोमेट्रियम रे. सेही जगह रे जदी मो ब्लास्टोसिस स्प्लिट हो जाउची इनटू टू डिफरेंट पार्ट्स, ताले कौन होबो ताले एठी अधा फाळे ह्यूमन बीइंग एठी आ अधा ह्यूमन बीइंग तयारी होबो कि एमती हुए नाही. एठी हुए ट्विन्स आर फॉर्म्ड. ताले ए जगह रे एमती केमती हला हाफ ऑफ माय सेल्स आर फॉर्मिंग वन इंडिविजुअल, half of the cells are forming a complete individual. ताले ए जगह रे एटा को हमें indeterminate एथि पई कहबा because these cells are not determined to produce a specific category of cells. That is why we say it is indeterminate cleavage. जेकोसी ब्लास्टोसिस स्टेज रू आमे जदि गोटे सेल भी आनी कि बाहर ग्रो करिबा, it will form a complete individual. That is because these cells are not determined to produce a particular kind of cells. They are indeterminate type. ताले हमें ह्यूमन बीइंग केस रे कहि परिबा it is indeterminate, holoblastic and rotational cleavage. Cleavage रे कौन होची ना the daughter cells that are produced are mitotically dividing rapidly without any cellular growth. आउ कौन होची ना those daughter cells that are formed we say those are called as blastomeres, and in this particular type of division there is a very short stretch of interphase. कहीं की ग्रोथ होनी. ए डायग्राम टी रे देखि परिबो आमे देखैचे दीटा डायग्राम जोथरे सेल साइकिल रे क्लीवेज केमती होची आ नॉर्मल सेल्स रे क्लीवेज केमती होची आमे देखि परिबा. जेतबेले सेल साइकिल क्लीवेज रे ऑपरेट करूची से जगह रे पिला माने देखि परिबो खाली दीटा फेज अछि, गोटे माइटोटिक फेज आ गोटे तमरो सिंथेटिक फेज एम फेज आ एस फेज. किंतु नॉर्मली जेतबेले सेल्स डिवाइड करे सेतबेले तमरो G1 S G2 आ एम फेज भी रुहे. क्लीवेज रे जेहेतु हमर ग्रोथ फेज गुडा को नाही तेणु क्लीवेज रे हमर सेलुलर ग्रोथ हुए नाही. अप टू द फॉर्मेशन ऑफ ब्लास्टुला there is no growth of the cells. Now coming to the entire picture, entire scenario. ए डायग्राम टी रे पिला माने देखि पारुथिबो starting from the formation of zygote to a 2 cell stage, तापरे 4 cell stage, then we have a 8 cell stage and then we have a 16 cell stage जोटा की लिखा है morula बोली. Now this you can see very well that these all are packed very compactly and thereafter after that the blastocyst formation takes place. केमती एगुड़ा को हुए हमें details रे देखिबा. early blastocyst आसीबो, then it will get implanted and then it will form the placenta and finally the embryo will derive its nutrition from the maternal endometrium. এই diagram টিরে देखिबो cleavage ର different stages. Once it has formed the blastula, morula आमे previous figure रे देखो एठी morula is a compact solid cell, ball of cells. But here in this figure you can see blastula is a hollow ball of cells. मझिरे गोटे cavity formation हुए जोटा को आमे कहू blastocell बोली. ताहले ये जो entire cleavage रो procedure यारो significance कौन कहीं कि it is more important for the embryonic development so that we can get a large number of cells that can undergo differentiation and gastrulation to form the different organs of your body. Now, one more aspect is जदि गोटे बड़ सेल अच्छी जो थर साइटोप्लाज्मिक कंटेंट बहुत अधिका आ गोटे छोटो न्यूक्लियस अच्छी ताहले दैट पर्टिकुलर न्यूक्लियस कैनॉट डू और कैनॉट वर्क द सिंथेसिस मेकानिज्म ऑफ द सेल विल नॉट फंक्शन दैट वेल. जदि आमे से सेल टी को छोटो छोटो parts रे divide कर दबा and each part we will provide with one part of the nucleus, then the synthesis machinery will function properly. सेइथि पई जते बळे cleavage होउची there is a increase in your nucleus to cytoplasmic ratio and this will help not only it will meet the transcriptional needs of the cell but this will also help the cell to develop properly. Now coming to the compact stage, आमे देखिले zygote to 2 celled, then 4 celled, then 8 celled stage division हैला, then we are getting a 16 celled morula. This morula is a compact unit. सब गुड़ा का cell बहुत compactly associated है कि थांती. So we also say this as a compaction stage. After the third cleavage, they compactly arrange. Third cleavage माने कोटा आमर फर्स्ट क्लीवेज है जोठी दीटा सेल फॉर्मेशन है. सेकेंड क्लीवेज इज व्हेन 4 सेल्स आर फॉर्म. थर्ड इज व्हेन 8 आर फॉर्म. जितेबाला दीटा जाक प्लेन रे डिवीजन होची आफ्टर द 8 सेल स्टेज कमिंग टू द 16 सेल पैकिंग इट इज वेरी कॉम्पैक्टली अरेंज. द जाइगोट reaches the morula stage when it is comprised of 16 to 32 cells and this morula will slowly come down in the uterine cavity. तलकू खसी आसीबो फैलोपियन ट्यूब रे खसी खसी के आसिकी यूटराइन कैविटी रे लॉज हबो एंड जेतेबेले सी ऑलरेडी 16 रो 32 सेल हे गलानी ता पाखरे आउ सफिशिएंट न्यूट्रिशन नाही. सेथि पई सी एंडोमेट्रियल लाइनिंग रो जो सिक्रीशंस हुए जहा को कि हमें कहू यूटराइन मिल्क, we can say it derives its energy from that or nutrient from that uterine milk. Now the fluid will begin to accumulate in the morula. धीरे धीरे fluid accumulation हबो आउ सेल गुड़ा को गोटे साइड को शिफ्ट हे कि जीवो. Now this morula will now be called as a blastocyst. ए जो डायग्राम टेरे देखि पारुथिबो morula सेल जेतबेले फॉर्मेशन हे जाउची पूरा कॉम्पैक्टली अरेंज हे कि अछि ता पाखरे म गोटे देछि मलबेरी रो डायग्राम. It is also said that it is compactly arranged just similar to that of mulberry. So we say that as morula. Okay. Now the blastocyst will have two different shell layers, outer trophoblast layer and the inner embryoblast layer. A diagram tere dekhi paribo. The outer trophoblast layer which is marked in single cellular structure tome dekhi parthubo, it will provide nourishment and it will form the future placenta. Chorion formation karibo and this will form the embryonic part of your placenta. आउ कौन देखि पारछो डायग्राम टिरे ना मझिरे गोटे hollow cavity like formation हेछि which is called as your blastocell cavity, the fluid filled blastocell cavity that has formed due to the incoming or the secretion from the endometrial lining that pushes the cell to one side and those cells will form the embryoblast or the inner cellular mass will form the embryoblast. मझिरे रहला तमरो blastocell cavity and throughout it is covered with trophoblast cells. Trophoblast will provide nutrient to the growing embryo and this inner cellular mass will form the embryoblast. And the inner cellular mass of the embryoblast that are closely in opposition to the trophoblast of that side, they have secreting cells which are called as cells of Rober. Okay. These cells of Rober are a thin layer of trophoblast that covers the inner cell mass. आउ जो पट साइड आउ गोटे जिनस तुम मैंने देखि पारथिबो ब्लू कलर लाइनिंग रे जोटा मार्क करा हैचे दैट इज द जोना पेलुसिडा माने तमरो एम्ब्रियो रो जोना पेलुसिडा लेयर इज स्टिल इंटैक्ट till the formation of blastocyst. Now we will have a short video to see how these things take place in the uterine cavity. Iti dekhi paruthibo, this is a egg or a zygote that will undergo mitotic divisions to form your morula. धीरे धीरे fallopian tube रे तल को आसुची 2 cell, then at 30 hours it has formed 2 cell stage. These are your blastomeres. They will go down to the fallopian tube, they will form the morula, and finally this morula will be converted to blastocyst. ताहले आमरो blastocyst formation हे गला. Once the blastocyst has been formed, next it will proceed to implantation. तेणु एतिकी जाए तो आमर जितेबाला एम्ब्रियोनिक डेवलपमेंट रे आमे कंटिन्यू करूचे ब्लास्टोसिस्ट वंस इट इज फॉर्म्ड जोना पेलुसिडा लेयर धीरे धीरे डिसइंटीग्रेट करिवो एंड नेक्स्ट इट विल गो फॉर इम्प्लांटेशन. ए पोर्शन रो बहुत क्वेश्चंस आसे एग्जामिनेशंस रे मोस्टली एमसीक्यूज. सो पिला माने टिके भलरे प्रिपेयर करिवो एंड गो थ्रू द डिटेल्स ऑफ दिस पोर्शंस. मोस्ट ऑफ द एमसीक्यूज कम्स in reproduction from these parts, your fertilization, implantation, embryonic development. Last class रे हमें up to blastocyst formation पढ़ी थिले. Blastocyst आसिकी endometrial lining पाखरे lodge हबा परे the process that will start is called as implantation. So we can define it as the attachment of the embryo after the blastocyst stage into the endometrial lining of the uterine cavity is called as implantation. Implantation takes place roughly around 7th to 8th day after fertilization. The blastocyst that has already been formed comprises of 2 layers, as we had already discussed, the trophoblast and the embryoblast. The trophoblast or the trophoectodermal cells will secrete enzymes that will digest your zona pellucida. Karana je parjanta zona pellucida intact achi, blastocyst will not be able to lodge into the endometrial lining. So in a way we can say that this zona pellucida prevents the improper attachment of the blastocyst or the improper implantation is avoided to a greater extent with the help of zona pellucida. Now, the blastocyst after hatching out of the zona pellucida is called, the process is called as hatching. Once hatching has taken place, the blastocyst will increase 2 to 3 folds in size and it will form a embryonic disc. जो सेल एम्ब्रियोब्लास्ट सेल फॉर्मेशन हेतिला और ट्रोफोब्लास्ट लेयर फॉर्मेशन हेतिला तमरा एम्ब्रियोब्लास्ट सेल्स विल गिव राइज टू द एम्ब्रियोनिक डिस्क. तापरे कोन हबो ना देयर आर डिफरेंट स्टेजेस ऑफ इम्प्लांटेशन. केमती ब्लास्टोसिस्ट आसिकी मनकु तो लागि जीवनी एंडोमेट्रियल लाइनिंग सहित देयर आर सर्टेन स्टेजेस थ्रू व्हिच इट विल firmly adhere to the uterine endometrium. The first is apposition, then adhesion with the help of integrin binding proteins, then invasion or migration or interdigitation, and finally it will be lodged into the completely lodged into the endometrial lining. आमे details रे देखिबा केमती theory होची तीनटा जाक process केमती होची simultaneously, step by step how it is taking place and how finally the blastocyst is embedded in the endometrial lining. The zona pellucida, as I have already told you, prevents abnormal implantation, and the endometrium is prepared for implantation by the complex activity of different factors. It is a combination of factors. Those are cytokines, growth factors, lipids, and all these are modulated by the progesterone. Now, how this bilaminar embryonic disc is formed? After 8 days after fertilization, your embryoblast will differentiate to form two different cellular layers, hypoblast and epiblast. Hypoblast are a flattened cuboidal cellular layer which will form your primitive endoderm, and epiblast is the external cell layer that is formed from the embryoblast cells, and these are tall columnar cells and they will form the primitive ectoderm. ये डायग्राम टी पिला माने देखि पारुथबो जहाँ जहाँ हमें डिस्कशन कले we have the trophoblast layer in pink colored flattened cells you have seen, and we have the embryoblast, then we have the maternal endometrial lining, and this is a diagram which shows how the blastocyst goes and embeds into uterus. एटा को हमें आउ टिके details रे discussion करिबा. So, here in this figure you can see maternal blood vessels are there, endometrial lining रे obviously maternal blood vessels रहिबो. तापरे तमर जो सेलटा आसिकी एम्बेड हेची सेतरे कोन चेंजेस होची, दीटा कैविटी स्ट्रक्चर एटी देखि पारुछ, गोटे ब्लू कलर रे अछि, गोटे येलो कलर रे अछि, एगुडा को कोन केमती फॉर्मेशन हुए, all this we will discuss in details. We will draw one single diagram, and with the help of that single diagram we will understand the entire mechanism. So, let me first draw the uterine endometrial lining. This is the uterine endometrium, or the place where your implantation will take place. These are the epithelial cell layers, and it is filled with maternal blood capillaries. There are blood capillaries, and this is the maternal blood capillary. कौन हबो तापरे हमरो blastocyst formation हे सारी थिला. Now, our blastocyst has two different kind of cells. We have an outer trophoblast and an inner embryoblast. तेणु एटा हला हमरो embryoblast, and the outer layer which is formed of flat cells, this will form my trophoblast layer, and we have a cavity which is your blastocell. Now, this blastocell cavity separates your inner cellular mass to one side, and this side which faces your uterine endometrium is called as your animal pole or embryonic pole, and just the opposite side is called as your abembryonic pole. ताहले embryonic pole कोटी ना जोटी मोर embryoblast cells रहिला, that part is called as my animal pole or embryonic pole. Just opposite to the embryonic pole is my abembryonic pole. तापरे कोन हबो मु कहितली जोना पेलुसिडा जे पर्यंत इंटैक्ट अछि, it cannot lodge into the uterine endometrium. So, first of all the zona pellucida will start to disintegrate, and this process is called as hatching. Once my zona pellucida has been completely removed, now my blastocyst is ready to be implanted into the uterine endometrium. एटी कौन हबो ना, the trophoblast layer will form two different type of cells, that are your syncytiotrophoblasts that will merge with syncytiotrophoblasts and cytotrophoblast. The syncytiotrophoblasts will merge with the uterine endometrium and the cytotrophoblast will remain as such. What will happen to the embryoblast? These will form a bilaminar disc. This is my embryoblast, these cells are my embryoblast cells. These cells, they will give rise to a bilaminar disc. A bilaminar disc which is composed of elongated columnar cells, which is my epiblast, and a flat layer of cells which will form my hypoblast. Okay. So, this will form the epiblast and hypoblast are formed. Syncytiotrophoblasts will merge into the maternal endometrium. Cytotrophoblast will remain as such. This entire lining will provide nourishment to the growing embryo. The cell will go, first it will oppose, come closer to the endometrial lining. Then there will be intracellular matrix protein that will help in adhering to the endometrial lining, and then after adhering, this particular embryo will invade, invasion will take place, it will invade and finally it will be lodged into the endometrium lining. So, this is how the entire process of implantation will take place. I hope all of you have understood clearly till now. তাহলে আমি দেখিলে the blastocyst has been embedded into the uterine endometrium. Amara implantation has been successfully achieved. Thereafter, after implantation, what will happen? The entire embryo will develop, get nourishment from the maternal blood supply, grow in size and finally develop into a complete individual. Now, what is decidua? Decidua is that part of the endometrium which is shed along with the fetus after delivery. Tamane, after implantation, the modifications that are formed within the endometrial lining in the uterine cavity is called as decidua, and this comprises of three types. We have decidua basalis, we have decidua capsularis and we have decidua parietalis, all depending on the place or the positioning that they are associated with the fetus. Now, we come to amniotic cavity. We have seen that the hypoblast cells and the epiblast cells were formed in the germinal bilaminar germinal disc. Now, these epiblast cells will develop a small cavity and this cavity will be called as our amniotic cavity. As this cavity will enlarge, a single layer of squamous cells will form a dome like roof, and this roof above the epiblast will be called the amnion. The amnion will eventually surround the entire embryo, creating the fluid filled amniotic cavity. Now, in this diagram, as you can see, the first diagram, the uterus has already embedded your embryo and the amniotic cavity has started forming. The blue colored cavity that is formed in between the trophoblast and the epiblast layer is called as your amniotic cavity, and these are the blood filled spaces of the maternal endometrium. The chorionic cavity has already started to form. The chorionic villi that are the interdigitations of your trophoblast layer into the maternal blood will form your chorionic villi, and this will lead to the formation of chorionic cavity further. And in the third diagram, you can see the yolk sac is formed, the chorionic cavity is distinct and the amniotic cavity is also formed. Now, what is extra embryonic coelom? The cells of the trophoblast will give rise to mass of cells that are called as extra embryonic coelom. These extra embryonic coelom is differentiated into the outer somatopleuric and the inner splanchnopleuric extra embryonic mesoderm, and both these layers will enclose the embryonic coelom. Now, in this diagram, as you can see, the trophoblast layer will form the extraembryonic mesoderm that will be differentiated into the inner and outer mesoderm, and in between there will be the coelomic cavity. Now, coming to the extraembryonic membranes, we have 4 extraembryonic membranes. They are your yolk sac, amnion, chorion and allantois. Now, coming to chorion and amnion. The chorion is formed by the somatopleuric extraembryonic mesoderm inside and the trophoblast outside. This will form a type of interdigitation, and it will also form the chorionic villi during the process of implantation. Now, the amnion is formed by the amniogenic cells inside and somatopleuric extraembryonic mesoderm outside. Later on, the chorion will become the main embryonic part of the placenta, and the chorion will secrete beta HCG. What this beta HCG will do? This will indicate the corpus luteum to keep secreting progesterone, which is the main pregnancy hormone. It maintains the endometrial lining. So, this beta HCG will help in maintaining the pregnancy further. Now, the amnion surrounds the embryo, creating the amniotic cavity that is filled with amniotic fluid. What this fluid will do? This fluid will act as a shock absorber for the fetus. It will regulate the body temperature of the fetus, and it will prevent any damage or further desiccation of the fetus. Yolk sac and primitive streak. Now, the flattened cells arising from the hypoblast will spread and line inside the blastocele. These are endodermal cells lining the primary yolk sac. With the appearance of the extraembryonic mesoderm and later of the extraembryonic coelom, the yolk sac becomes much smaller, and now it is called as the secondary yolk sac. The change in size of this yolk sac is due to the change in nature of the lining cells. The lining cells are no longer flat, but now they have become cubical. The secondary yolk sac consists of outer splanchnopleuric extraembryonic mesoderm and the inner endodermal cells. The yolk sac is a source of blood cell, and it functions as a shock absorber and helps prevent desiccation of the embryo. Now, in this diagram, you can see the amniotic cavity, yolk sac formation, the extraembryonic coelom being formed, and the primitive streak is appearing. And in the adjacent diagram, you can see a transverse section in which the ectodermal layer, endodermal layer and mesodermal layer in the process of formation, that is the 3 germinal layers, are starting to form. Now, we will come to gastrulation. Now, after implantation, what will happen? The first major event that will take place in the 3rd week of development is gastrulation. We had a bilaminar disc. Now, this bilaminar disc will be converted into a trilaminar embryonic disc through this process. There will be rearrangement and migration of cells from the epiblast, and it will also be followed by the formation of primitive streak. Now, what will happen? The gastrulation involves the rearrangement and migration of the cells from the epiblast. As we have already discussed, the primitive streak, which is a faint groove on the dorsal surface of the epiblast, is formed. It will appear as a faint groove on the dorsal surface of the epiblast. It elongates from the posterior to the anterior part of the entire embryo. The caudal end and the anterior end are marked, as well as the right and left sides are demarcated at this particular stage. Now, coming to the formation of germ layers or embryonic layers. After the formation of the primitive streak, the cells of the epiblast will move inwards below the primitive streak, and they will detach from the epiblast layer. This inversion that occurs, this inversion is called as invagination, and this will lead to the formation of your mesodermal layer. This invagination will form the all the 3 distinctive embryonic layers, that are your ectoderm, mesoderm and the endoderm. Once these cells have invaginated, some of them will displace the hypoblast, forming the endoderm. Endoderm will develop first during the embryonic development, and then after that the other cells that will remain between the epiblast and newly formed endoderm, they will form the mesodermal layers, and the remaining cells of the epiblast will form the ectodermal layer. In this diagram, we can have a better understanding of whatever we have discussed. The primitive streak formation takes place. There is a groove like structure in the middle, you can see. This blue color layer is your epiblast layer. This epiblast layer invaginates and moves up. So, there has been a rearrangement of cells. Now, this leads to, you can see the dispersal of cells below the invaginating epidermal layer or the epiblast layer will form your mesodermal layer that are marked in red color cells. These red color cells will form your mesodermal layer. So, subsequently, the hypoblast cells are also displaced and the definitive endoderm is formed by the dispersing cells from the epiblast. So, your epiblast is forming the distinctive ectoderm that is marked in blue, mesoderm that is marked in red, and the orange color layer beneath will form the distinctive endodermal layer. Now, these three germinal layers are formed completely. Now, let us take a look at this particular flow chart, wherein we can discuss which type of cells are derived from which particular layer. A blastocyst will comprise of two cellular layers, trophoblast and embryoblast. Embryoblast will contain the inner cell mass. The trophoblast will form syncytiotropoblast and cytotrophoblast. The inner cell mass will be further divided into hypoblast and epiblast layers. The hypoblast will form the extraembryonic endoderm, and this will lead to the formation of yolk sac and finally our extraembryonic mesodermal layer, whereas the epiblast will form the amniotic ectoderm and the embryonic epiblast. This embryonic epiblast will give rise to primitive streak as well as to the embryonic ectoderm, and the primitive streak formation will lead to further development of embryonic mesoderm as well as the embryonic endodermal layers. Now, what is the fate of the three germ layers that are formed? Each germ layer gives rise to specific tissues, organs and organ systems, and they have specified functions. The endoderm layer becomes digestive system, liver, pancreas and lungs. The mesodermal layer becomes circulatory system, lungs, skeletal system and muscular system, and the ectodermal layer forms the nervous system, skin, hair and nails. Now, let us see a video that will give a descriptive account of whatever we have already discussed. Now, as you can see, this is the bilaminar embryonic disc formed of epiblast and hypoblast cells. It has an amniotic cavity. The upper cells will form the amniogenic cells that will secrete the amniotic fluid, and it will form the amniotic cavity. Now, the primitive streak forms, the caudal end is marked and the head end is also marked. This primitive streak formation occurs at day 15. At this stage, the formation of this primitive streak defines the cranial end and the caudal end. As you can see, the left and right side of the embryo are also distinctly formed. Now, there is a primitive node formation which contains a circular depression which is known as primitive pit. This depression continues along the midline of the epiblast towards the caudal end of the streak. Our primitive groove has already been formed. Now, this process is called as invagination. We have already discussed epiblast slips into the embryo. The first cells to invaginate through the primitive groove invade the hypoblast and they will displace its cells. The hypoblast cells are eventually completely replaced and this will form a new definitive endodermal layer. By day 16, the majority of the hypoblast has been replaced and the remaining cells of the epiblast are now referred to as ectoderm, lined or marked in blue and forms the exterior distal layer. Some of the invaginated epiblast cells remain in the space between the ectoderm and they will form the newly definitive mesodermal layer. Those are shown in red color. Now, the 3 germ layers are formed: ectoderm, middle mesoderm and endoderm. Now, no more invagination takes place. Throughout the gastrulation, the ectoderm will continue to form the cranial end and the caudal end and it will establish the 3 germ layers. And this will complete our process of gastrulation. So, finally, all the 3 germ layers are formed and our cell is now ready for further development. So, this completes our gastrulation and I hope you all have clearly understood the process, how implantation takes place, how gastrulation takes place, how the germ layers are formed, and I hope you will all be able to answer whatever questions will be asked from these portions. Thank you.